today. We are going to have a great session. We have 3 speakers today who will be giving us some great perspective on the lessons learned and results of the recently presented EUCLID trial results. Just a brief introduction of our 3 speakers. Many of you know them already, I'm sure. First, we'll be hearing from Dr. Manesh Patel, who's Associate Professor of Medicine here at Duke. He is the recently named Chief of the Division of Cardiology and the Co-Director of the Duke Heart Center. I've also been told interesting fact that he moonlights at Wake Med under a different name. This hasn't been confirmed, so if there is additional information, Dr. Raj, okay, so it has been confirmed, so thank you for that. Dr. Jones, Schuyler Jones, is our second speaker. He's Associate Professor of Medicine here at Duke and Medical Director of the Duke Cardiac Catheterization Laboratory. There were a couple of interesting facts on his CV I wanted to mention, but he told me the only really important one to mention is that he's from Arkansas. So wanted to be clear on that. And then finally, we'll be hearing from Criag Reist, who's Assistant Director of Megatrials here. He has his PhD in biochemistry from UVA and has actually been with the Megatrials group for 18 years. And another interesting fact is that Dr. Reist's first day here with the Megatrials group was actually Schuyler Jones' first day of high school. So they have that in common, and just thought I'd mention that. Close. Monday versus Wednesday, I think, right? So thank you so much to our speakers for being here. Thank you to you all for being here, and I'll turn it over to Dr. Patel.
That was a great intro, and I was most excited to learn Schuyler went to high school, so that's really good in itself. In Arkansas. So I will try to use this microphone, and again, it's a pleasure to give DCRI Research Conference. I'll just start by saying, whenever you give research conference, especially around a trial like EUCLID, and lessons learned, obviously Craig, Schuyler, and myself are representing many hundreds of people here that have worked on the trial. And so when we share lessons learned, we want to also just make sure we thank everyone who worked on such a tremendous effort. So our outline is hopefully here. I'm going to talk about the unmet need of peripheral artery disease, and then Craig's going to talk about the actual trial conduct as it was. Schuyler helped throughout the trial conduct, but is going to talk with a little bit of specificity around the CEC process. I'm going to share the results that we shared at AHA, just so everybody's seen those results, and sort of talk about what does that mean, and then think a little bit about lessons learned for overall trials in PAD. So why study PAD? So some of you have seen us show this. This is work from the DCRI. This is the amputation map of the United States, unfortunately. And so we're in North Carolina, where patients are more likely to have peripheral artery disease and more likely to have a lower extremity that is unfortunately amputated. We're also in the stroke belt. We're also in the hypertension belt. And so one of the things we were, as interested vascular medicine physicians, wondering how do we help take better care of these patients? Schuyler, Shriek, Leslie, others have highlighted that the Medicare mortality is 48% at 1 year after a potential limb loss. And then Shriek and others have highlighted that up to 30-40% of these patients sometimes don't get any arterial testing, ultrasound, or ABI before they might have damage to the lower extremity. So we often say that patients know to come immediately in to see their physicians if they have pain in their chest, but when they're walking, if they have pain in their calf, they don't know that that might be a risk equivalent. And so as a clinician who's taking care of vascular disease broadly, we were very interested in this area, and we started doing these outcomes towards the studies to say how did they do. Some of you might remember Samit Subarwal. He worked in our group with others, and Eric and Emil Fosbal and others, and we said actually in the Danish registry, we're able to say what is the amount of sort of the evidence-based therapy, antiplatelet therapy used for patients. And you can see here that if you had coronary artery disease alone, you got a certain number of patients got antiplatelet therapy. If you have PAD plus CAD, it was less, but towards CAD alone. And if you had PAD alone, the utilization of antiplatelet therapy in these patients was pretty low. So even recognition amongst physicians, and this is a place where aspirin prescriptions are known, to use even aspirin was not well known. And so we were left with this perspective of what is the comparative effectiveness of therapies. We did that sort of AHRQ-funded research. And we were left with saying there's a huge unmet need. About 8 million people in the United States have peripheral artery disease, probably 200 million people worldwide. It's a coronary artery disease equivalent. We've done lots of trials on patients with coronary disease. We've done limited number of trials in patients with peripheral artery disease. Yet we take the evidence from one group and apply it to the other, and how do we do? So how about locally? So Schuyler and I and others developed maps like this. We've shown these at other conferences. This is now the North Carolina state by county, and how you're likely to potentially have a risk of losing a limb based on where you live. And that's, of course, a socioeconomic status issue, access to care, several other things. But as Duke Heart and Duke Health, we have a lot of hospitals that we care for and partner with. And so Henderson's a place we go do outreach to in Parham and things like that. So in the backdrop of understanding these patients in our own community and worldwide with PAD, we looked to the guidelines, and the guidelines said that for peripheral artery disease, you should use an antiplatelet agent. Didn't say which one, aspirin or clopidogrel or something. And that was a class 1 recommendation. And so we were able to go and look and say, well, the existing evidence is old. We want to study a new therapeutic called ticagrelor, and we'll get into the background of that in a second. But we endeavored in about 2011, 2012 to study one antiplatelet agent versus another in patients with peripheral artery disease. So as a sort of hope of background, this is, I think, hopefully a story that I think DCRI is best at, which is there are patients, clinicians, people working in a field where they see patients that need some new evidence and new information, passionate about figuring out how to do it, going to our industry sponsors or others and saying, how do we get patients or information in this area? And then we tie them to a wonderful organization to say, can we answer that question? So with that, I'm going to hand it to Craig, who's going to tell us how we tried to answer that question.
Thanks, Manesh. So I'm going to talk a little bit about some of the operations, some of the milestones in Euclid, and I've got about 15 to 20 minutes because Schuyler's got a patient on the table and he's got to get there, and I want him to be able to talk to. That or the Korean barbecue trucks out there, and I know he's a big fan of their dumplings, so it's one or the other. So how did we get here in Euclid? Euclid was a bit of a long journey. In 2010, there was a trial called Plato that was published, and based on the outcomes of the PAD sort of sub-cohort of patients there, it led to this concept of, should we do a PAD trial with ticagrelor to see if it's better than clopidogrel? So in early 2011, we began a partnership with AstraZeneca, DCRI, and the Colorado Prevention Center, which is Will Hyatt's group out in Denver, to see if we could operationalize what would then become the largest trial in PAD. Midway through 2011, we did our country selection, site identification. Later that year, the trial officially became named Euclid and became one of the AstraZeneca Parthenon life cycle development plans for ticagrelor. Those of you that worked here at DCRI know that we did a trial called Socrates as well, which was a trial in stroke patients, and there's still a couple of other ongoing trials that AstraZeneca is doing to look at ticagrelor in different patient populations. End of October, study leadership met with the FDA to discuss the trial design, and then in early 2012, protocol, case report form, endpoint definitions, and those things were finalized. Sort of the end of 2012, fall, November, December, we had our investigator meetings, first patient enrolled in Sweden, and then actually on New Year's Eve, 2012, first patient was randomized in the United States into the Euclid trial. Early 2013, we had additional investigator meetings, changed the sample size based on event rate estimates. Manesh will talk a little bit about some challenges there later in the presentation. And then in March of 2014, our last patient was put into the trial to reach our goal of 13,885 patients. We locked the database on September 27th earlier this year, and the results were presented at AHA in November. I'd like to show this slide because we like to brag a little bit about our ability to put patients into trials. And if you look at the blue line, that was our original enrollment projection. We exceeded that by quite a bit there, and even with our revised projection to get to 13,500, you'll see the red dotted line. We were able to achieve that slightly ahead of schedule. So a great effort by all of our site investigators, all the operational colleagues to put patients into Euclid at a very rapid rate. Just some of the numbers from a trial this big. You can imagine there's a ton of work that goes into getting patients into the trial. We actually had to enroll 16,445 patients to get to our 13,885 randomized patients. Had over 400,000 patient visits that were completed, almost 2 million case report forms, 1.5 million queries, about 12,000 CEC triggers, and 9,000 SAEs, so a ton of work and a ton of effort from a lot of the folks in the room here. So one of the things we like to do in addition to looking at the clinical trial results is look at how we did operationally. So you've all heard the term evidence-based medicine. We sort of piggyback this concept of evidence-based operations on top of that. And that's what can we learn from the last large mega trial that we've done to incorporate into the current trial. And then based on what we've done in the current trial, how can we move that into the next trial. So I'm going to highlight some of the sort of lessons learned from an operational perspective here. I just picked a few things listed on the slide. Schuyler's going to talk a little bit about the CEC quality plan that was a big part of EUCLID. So some of the things that we do in terms of how we define quality are based on a set of guiding principles. And this slide's a little different than my normal slide. One of the things you'll learn when working with Manesh is you send him something and you just never know what you're going to get back. So my guiding principle slide turned into this concept of randomized clinical trials being like an ocean liner.
Can you just stop and say that it was the Titanic? It was always the Titanic. And I was like, the Titanic's sake. Did I stop doing
That's right. We don't want that analogy. But I can pick up what he's laying down here. So the ocean liner concept is you got to build it right. You got to make sure you get the right patients in the trial. Then you got to make sure that as you're taking your voyage out to sea that the patients are treated in the right way so they get the right drug, they stay on drug as much as possible. And then eventually you got to bring that big ship into port. And so this concept of making sure that we get the right data, the right safety and efficacy events, that it's complete ascertainment of our primary endpoint data. So all that goes into defining quality and getting the right answer at the end of the day. So some of the way that you need to get there is through picking the right sites or getting the right crew on board your ship. One of the challenges we ran into with EUCLID early is we really didn't have a PAD network of sites. As I mentioned, there was a lot of PAD patients in some of our CAD trials, but we didn't really know who were the right sites to go to. Is it vascular sites? Is it cardiologists who maybe see PAD patients? So we did something a little bit unique in that first we partnered with a group, Colorado Prevention Center, who did a lot of these trials, and they were able to provide us with some sites. But we also set up a booth at the vascular interventional meeting out in Las Vegas in October of 2011. Schuyler, myself, Danny Underwood, one of my colleagues, went out there, and we actually recruited investigators at a clinical meeting.
To make the Las Vegas trip. I didn't get it, but
Bern was there.
That's right. So in addition to teaching Danny how to play video poker, we actually recruited quite a few good sites in a somewhat unique way. So when you think of operations, one of the things you think of is monitoring. You've probably all heard this concept of risk-based monitoring, and what that means is sort of open to a lot of interpretation. But at the heart of it is looking at the data and doing source data verification. Well, source data verification is essentially going out, having a clinical person look at the data in the case report form. Does it match the data that's in the hospital source document? And there's varying strategies on how to do this. The old school strategy is kind of a carpet bombing approach where you go out and you look at everything and you make sure it's right. Well, where we've tried to evolve is, do you really need to do that, or can we target what we're looking at to make sure that the important things are there, and also do a random sample of some of the other data to make sure it's okay too. So we sort of coined this concept of source data review, which isn't complete source data verification, but targeted source data verification. So the areas of the case report form that we looked at were the baseline hospitalization, because we want to make sure that the right patients got in the trial. So looking at the inclusion-exclusion criteria to make sure that the sites were following the protocol and enrolling the right patients. We targeted our hospitalization visits and follow-up because that's where a majority of our safety and efficacy endpoints are going to be associated with a patient being rehospitalized. So we spent a lot of time and we looked at 90% of our rehospitalizations to make sure that that data was accurate and correct. And then, as I mentioned, we also do a random sample just across the board of other follow-up visits to make sure that the sites are consistent in what they're capturing. So some of the findings of that on-site monitoring effort and looking at an SDV versus SDR approach, if you look at those outpatient visits, these are just the random sample of visits, and you take an SDR approach versus an SDV approach, you find about the same percentage of SAEs and endpoints that were missed by the site. If you target those hospitalizations, you get a much higher bang for your buck in terms of finding things that are missed. But again, looking targeted-wise at SDR, you find about the same amount of things that are missed. And then for the inclusion-exclusion criteria, again, a much larger bang for your buck with the SDR versus SDV in terms of the total numbers, percentage-wise about the same. Just an operations note, it takes about 2 to 3 times the effort to do complete SDV versus a targeted SDR approach. So it's more efficient, and we think it gives about the same level of quality. So that's something we're incorporating into a lot of our newer trials. In addition to having folks go out and do on-site source data review and monitoring, we did a lot of centralized review of the data. The stats team here at DCRI helped us generate reports to look at rates of endpoints or events. If you imagine, it's kind of like you miss the forest for the trees sometimes. If you're looking at things on site individually, you don't know that, hey, this site's enrolled 50 patients, but they don't have any events. That's probably not possible. They're probably missing something. So we had reports that allowed us to look across the data and identify patterns where we needed to focus some more effort and more attention on working with those sites to say, hey, are you really thoroughly looking through your medical records to make sure that you're not missing any serious adverse events or any potential endpoints. Same with rates of other things like study drug discontinuation, patient withdrawal. By looking at it across the board in a higher level statistical manner, you can identify patterns and then take action on that. We also had listings of things like patients on prohibited meds or patients where they didn't meet the entry criteria that allowed our in-house monitoring team to then quickly follow up with those sites, re-educate if necessary, and make sure that the data was captured accurately in the database. Some of the other things that we've learned from this is, if we can proactively identify the sites that are at risk of making errors and quickly work with them, we'll have a much better, much higher quality data set at the end of the day. Some of the sites that we've identified as high risk are those sites that are enrolling patients quickly, or those sites that have a lot of patients in the trial. Just volume in general tends to be an indicator that there might be some errors there, might need to put a little extra effort there. Sites that are on a corrective action plan, so if they've had an error or had an issue in the past, making sure that we follow up with those all the way through the end of the trial to ensure that that corrective action is maintained and staying in place. And then sites where there's turnover of key personnel. So if the study coordinator, for example, leaves and they have a new person, making sure we're going out, doing some retraining, following up to ensure that that person is well aware of what the study requirements are. So we've done a lot of additional on-site monitoring when those instances came up, and we've looked at a higher percentage of that data to make sure it's accurate. Some data cleaning innovations. A lot of this has to do with specifics of our data collection system. The Inform ECRF platform had a function that you could basically enter a comment on the page, and it would allow you to not have to enter any data in the form to look clean. So in these trials, it's a challenge because sites are trying to do the right thing, but often they'll take the easy way out. And so if they don't know something, you put a comment bubble in there that says, hey, I don't know this, as opposed to taking the extra effort to look for the data and really make sure that it's, if it's there, we want it there. So we disallowed the use of the comment bubble, so basically made that so it wasn't an option. So the sites were forced to really look for the data and work with us to make sure that they found it, whether it was looking at an external hospitalization or really digging deep to find the information and not just getting the get out of jail free card comment bubble of not being able to capture it. We did a tiered querying strategy where the data management group would post a query. If the site answered it and it wasn't an effective answer, they'd query it again. And based on how they labeled the queries, we could tell if there was a lot of ping-ponging back and forth. What we tend to get is we'll query the site for something and they'll say, well, the original value is correct. Well, it can't be because it's physiologically impossible to have a blood pressure of 1200. Probably 120. So those kind of things, to avoid some of that ping-ponging back and forth, we created a system where we could review how many queries are going back and forth several times and have a person actually take action on that and connect with the site. We had a remote drug accountability module. So one of the challenges in a trial like this is keeping up with all of the logistics of, study drug coming in, study drug being assigned to patients, and study drug being sent back to the site. So knowing that we're only going to send monitors out on site, in an infrequent basis, we wanted to make sure that we had people in-house that could follow up with that and not allow that to be a backlog that creates problems at the end of a trial. And then last on this list is free text review. We try to avoid free text in our case report forms, but there's always instances where we do have free text. That's led to some challenges in the past where sites tend to give you more information than perhaps you want in free text sections, and being able to look at that and determine, is this a serious adverse event or is this a potential endpoint that needs to be reported somewhere else in the case report form? We implemented a thorough free text review plan that allowed us to feel good at the end of the day that we were getting the right data in the right place. So what does the future look like for large trial operations? Some of the things I mentioned is just working to proactively identify those sites that are at risk throughout the trial and taking action to try to mitigate that risk. More rapid identification, escalation, action, and follow-up on issues that are impacting quality. Again, a lot of the things that I just mentioned. And then something that I encourage some of our DCRI operational leaders to get more involved in are talking with other thought leaders across academia, industry, and the regulatory agencies regarding how we define quality in large trials. Several of the DCRI leadership have been involved in the city effort, which was the group that sort of led the risk-based monitoring guideline that the FDA put out, but continuing to work because there's a lot of challenges that we still face. We went through a regulatory inspection here at DCRI. We've had several of our trials get inspected, and some of the things that we're trying to sort out are, when do you close a site for noncompliance versus trying to do everything you can to keep those patients followed and not have missing data. So there's always the challenge of keeping a site compliant, but then if you close them, you don't want to lose the opportunity to follow that patient and get complete ascertainment of your endpoints at the end of the day. So working together to sort of define what does quality in a large trial look like, how are we going to manage that, and how are we going to ensure at the end of the day that we have data that meets everybody's expectations. Schuyler.
Ready. All right. Thanks, Craig. So before we do CC, I thought I also helped run the helpline, and we had a great partnership with AstraZeneca, I think, between Brian Katona and their medical staff. We really did a great job with the helpline, taking calls. I actually can remember we hadn't gotten calls for like months. This was early. Was it earlier this year or last year? Craig took 3 calls. He was 3rd call on the list. And I get this email from Craig, and it was, "What the hell, man? Why are you answering the calls?" And Manesh hadn't answered. He was 2nd call. And I was in Costa Rica on spring break, and I was like, I don't think I get reception down here. But we had, all along, great partnership, great memories of EUCLID, and a lot of fun. I have a feeling when I leave, they're going to tell a lot of stories, so I want each of you to hold them accountable for much of what they're saying. So CC was busy. We started, like Craig said, in kind of 2011, 2012, ramping up. We started in DEP. Does everyone remember DEP? So we finished in EAM. I can remember, I actually looked for the text just now because I texted Manesh and Craig the day that DEP went away, and I said, "Ding dong, DEP is dead." And they were like, "What does that mean?" And I was like, "The whole thing's gone." So we had a rough start with DEP. We had multiple meetings where we couldn't process events. We ultimately converted to EAM, and then we really were consistently at 95% complete. Everybody remember what DEP is? Anybody not remember? So DEP is an electronic adjudication platform that the DCRI created, and it was not a success. And EAM is an electronic adjudication platform that is a success that we now use consistently throughout our trials. Sorry, I probably should have said that. So in addition for the CC, we did almost 12,000 events. We also added an endpoint in the middle of the trial, and that was acute limb ischemia. And we retrospectively queried sites and then reviewed each of those events, and then we prospectively did that throughout the trial. All right, so I thought we'd talk a little bit about the quality control process that we implemented, and this was pretty standard, and then some of the newer stuff that we thought about throughout the trial. So we actually did 3 separate quality control reviews throughout. We did an early one that was heavily weighted towards the primary composite event, just to make sure that we had death, MI, and stroke done correctly. We had a very few bleeding events that we did, but at that point I don't think ALI, Stacey, I don't think we did ALI then. So in the middle part of the trial, we had another bolus of events that we reviewed to look at both bleeds and acute limb ischemia, and saw that we were doing, despite 4 bleeding scores in Euclid that we used on our adjudication form, we were actually doing a very good job of adjudicating those events. And then at the end, there's this big discussion about what's enough for QC review for big trials like this. And at that point, I think we had done like 4% of the overall volume. So we said we need to do a bigger sample of QC. So we did 5% of the overall triggers at the end of the trial as well. And we really focused that on primary composite events. We were concerned about stroke as it had been adjudicated, and we ended up having no issues with that. So this also helped us identify and then re-review important endpoints that are tough to adjudicate. So type 2 MI, undetermined deaths, and then acute limb ischemia is tough because we had never adjudicated it before. And so we thought that that process helped us a bunch. So it was probably the last year of Euclid, the megatrial group and the CEC group really partnered. I think Euclid was the backbone or the structure that allowed us to partner so effectively to understand how can we get better as a CEC and a megatrial group. And so the quality plan that we'll just talk briefly about is really spawned from that. And so a lot of it was based on what we had learned from FDA reviews of other big mega trials. And so one of them was source data tracking, and then the sufficiency or insufficiency of source data as you adjudicate. And a lot of times those are really focused on what's called, the high-risk events. In many cases, it's negatively adjudicated events. So non-fatal events like MI and stroke that are said adjudicated as no event, or undetermined death. So you don't know what caused the death, and death is rarely negatively adjudicated, or in most cases we don't allow it to be negatively adjudicated. It either happens or it doesn't. So that was one of the kind of processes that we went through, and I think that helped us think through how do you determine when the reviewer had enough data to call an event, and then track it throughout the trial. One of the other things that we did, and this wasn't CEC specific, but we did free text reviews as the FDA was really concerned about what happens in narrative fields and other free text fields in big trials. Are you missing things? And so we did actually a pretty extensive evaluation of all the free text, pretty much every free text field that existed in our database, and that included hospitalization, lab fields, and then our narrative fields. And so that was, I would say, a lot of work. We did find a number of events, and we've figured out a way to implement this into other trials. And then for negatively adjudicated events, in addition to source data kind of insufficiency or sufficiency, we thought that it was important really to create a process, and actually we did this up front, and it helped us a lot, which was to put things into buckets. So when things get adjudicated, they're either positively adjudicated, or they're negatively adjudicated, or they're in this other disease category, or this other category that we specified, which was disease progression. Some of the CEC has seen same disease entity or similar pathology. So we think that actually is a pretty useful designation for big trials where you have a lot of events being reported to understand where your risk exists and how you can improve your process. So at the end of the trial, or nearing the end of the trial, we had a number of things that I was actually very comfortable with, but we had some questions about consistency and were we adjudicating things similarly across the trial. And so we did a number of stat checks. And in this process, I thought was really useful. It was time consuming, but we, I think, did a very nice job with that. We really focused on the events that occurred around deaths. And so if you look at kind of this example. It's every death with a known cause. We looked at it. We performed a stat check to determine if the corresponding event was adjudicated positively. So for an MI example, if a death occurs due to an MI, we wanted to make sure that there was a corresponding MI event adjudicated as fatal on the MI form. Hope that makes sense, but we did the same thing for MI, stroke and bleeding to ensure that there was consistency. What we didn't want to see was that there were a bunch of things that were called deaths due to MI, and they weren't deaths due to MI. We don't even have that many MIs in the database. So I think that that process we created about 6 months from database lock, and then we performed it 2 or 3 times as we moved towards database lock. All right, other checks, and these are, I think probably we can just briefly mention these. We looked at other things that we thought were probably higher risk things for CEC. So when you look at when a PI reported an MI and there were cardiac markers at meta definition, so 5 times the upper limit of normal, but we called it no MI. We thought that was a higher risk issue that we should probably look at again. So we looked at each of these. Same thing for stroke, except if they said that a stroke occurred and symptoms lasted longer than 24 hours, which should define a stroke, but we called it no stroke. We looked at all of those. Same thing for a lot of these things in terms of intracranial bleeds, things that really matter to patients, and then the FDA is very critical about, as well as hemoglobin drops that occur within a time frame. Remember that not always with CEC do you have all the data right in front of you. So with stats, you had all the data with hemoglobin drops and then cardiac markers, and you can really put it into perspective there. Last thing is that we did an independent review of the charter about midway or a little bit after midway through the process, trying to standardize terminology. That led us to creating our templates for the CEC charters, and I think that made our process better. We updated our QC plan. We updated the specifications of the electronic adjudication platform, and then how we were processing source documents. So we also modified our TEMI definition throughout, or at that point in the charter, and that was just an interpretation thing, allowed us to further define that bleeding process. Manesh, I think I'm turning it over to you here, and then I got to go back to the cath lab, so don't believe anything he says.
Thanks, Schuyler, and thank you all. I'll just do a little check. I mean, what an amazing journey. You're going to get to see the results here in a second, and I think hopefully what you've heard so far is a lot of how quote-unquote the sausage is made when you do these trials. I mean, how many patients we're looking for, the sites, what innovation we're trying to do. I think Craig and Schuyler have highlighted, and hopefully you'll see that a lot of this is teamwork and stuff we do here at the DCRI. So I had the opportunity to present the results. One of the things I'll just highlight for you that you maybe didn't see that I'll share stories about. So Craig told you that the database locked on the 26th or the 27th, and then we unblinded on the 30th. So that's 4 days from locking to unblinding, and that was September 30th, and then we presented the trial results at AHA on November 13th. So these are pretty compressed timelines with a lot of people working very, very hard at nights and weekends and different things to get this to happen. And so I want to again recognize all the work that happened in-house, but then also recognize all the people around the world that helped us do that. So 13,885 patients. I'll come back to this again. At this point in the world, a PAD trial had not been conducted with this number of patients. We were able to enroll 14,000 patients in 14 months. So as an organization, as a trial group, we have to think about how we do that. But we also showed you that we had 2 million data points, several thousands of SAEs on a drug that was the 4th large randomized trial. You might argue, what was new to be learned about the safety of the compounds? Clopidogrel had been on the market for 20 years. Ticagrelor had been on the market at least several years when we did this. Did it have to be blinded? I'd start asking these questions as lessons learned, because as Chris knows and others in this audience, we're trying to figure out how do we do these faster, better? How do we get more questions answered in ways for our patients? And so first, let me show you the results. It starts with the people that participated. These are the top 10 enrollers, and they're all from around the world. To get them off study drug and get their endpoints and SAEs and AEs identified. And you have to make sure that we're capturing data the same way. These are sort of the operational challenges you can think about. This is a thank you to all the site investigators, and they fit not on one slide, but two slides. It's an honor to stand up in front of everyone and share the results, no matter what the results are. I'll share the results now, but it was certainly an endeavor that you heard started in 2011. So 2016, after 5 years of building this boat, taking it out to the ocean, bringing it back and saying, did you stick to your principles? Did we enroll the right patients? Did they get treated in the right way? And did we count the right endpoints in the right manner? Think about that as I share the results with you. So these were the disclosures that it's important to recognize that, of course, EUCLID was sponsored by AstraZeneca. So I spoke a little bit about this, peripheral artery diseases, and we changed this word. After we saw the results, we changed this word. Every paper to date, before we wrote the paper at the end and did the trial, it said peripheral artery disease is a systemic manifestation of atherosclerosis. And we changed the word to add considered, because what we realized is we had always thought peripheral artery disease is the same as I just described it. You have chest pain, you have leg pain, they're the same disease. Well, the question is, are they the same disease? Are the endpoints driven by the same things? Is the biology the same in an outpatient? So there are 200 million people worldwide. Jerry Folks, one of our executive committee members, is an epidemiologist, and during the conduct of the study, he had written a paper in Lancet, which is referenced now, to talk about the scourge of peripheral artery disease around the world. PADs associated with both cardiovascular and limb morbidity and mortality. One of the things we were struggling as we designed the trial is to say, what should we measure? What do patients care about? Traditionally, FDA says, well, we care about cardiovascular death, MI, stroke. That's our usual endpoint trial. But patients care a lot about the ability to walk, pain in their leg, the actual loss of limb or acute limb ischemia. So we tried to incorporate that into the study, but it couldn't be part of the primary, at least at that time. That's changed for other trials, I think in part because of some of the conversation we had with EUCLID. Antiplatelet therapy is recommended for all patients. As I said in the guidelines, antiplatelet therapy is recommended. And in fact, we said, what's the comparator? So when we were designing the trial, we said, what should we compare the new potent antiplatelet therapy to? So this is the extent of at least the monotherapy data for peripheral artery disease. This is the Capri trial, published in 1996, now 20 years old, where they took a broad range of patients with atherosclerosis and randomized committee members wanted. I can tell you, hopefully in the future, we've designed systems where we can answer those clinical questions faster. One, to put to bed how good aspirin is. Two, to inform our practice better. But that being said, the background for the time of Euclid was PAD is common, causes high-risk cardiovascular events. Clopidogrel is felt to be superior to aspirin in PAD patients. Ticagrelor, therefore, was an antiplatelet agent, felt to be potent, and it reduces cardiovascular death, MI, and stroke in patients with, compared to clopidogrel in acute coronary syndromes, the Plato trial, and it had been proven to be an effective chronic therapy in patients with a prior MI. Now, I'll remind you, in that trial, it was ticagrelor plus aspirin versus aspirin, so it's a 2 versus 1 question, but it had been shown to be better. So the primary objective of Euclid was to determine if long-term monotherapy with ticagrelor compared to clopidogrel reduced cardiovascular death, MI, or ischemic stroke. The secondary endpoint included acute limb ischemia. This is the design of Euclid, and this took us about 6 to 8 months with the FDA. Why did it take us so long? Well, so we knew we were going to test ticagrelor versus clopidogrel, but clopidogrel was becoming generic. So one of the questions was, are you going to use generic clopidogrel, or are you going to go get what I'll call store-bought clopidogrel from Sanofi? And thankfully, the sponsors were like, because the FDA wanted, we went and got not a generic that's on the market, but the store-bought clopidogrel from Sanofi. We said, well, who do we want to enroll? We want to enroll symptomatic patients with PAD. So the ankle brachial index, I tell patients, if your blood pressure is 100 in your arm, it should be 100 in your leg. If it's 50 in your leg, 50% of the blood flow is not getting to your leg. So your ankle brachial index is 0.5, and there's a risk associated with not having 50% of your blood flow be blocked somewhere between your arm and your leg. So that was one of the inclusion criteria. Pain on some sort of ambulation, typical or atypical, and a lower ankle brachial index. We can actually use the ankle brachial index, and it's associated with limb and mortality because the lower it gets, the more atherosclerosis people have. So it's 0.8 or less, or 0.85 at 2 visits, and then a prior revascularization. Now, this was a stickling point that the FDA really pushed on. They wanted us to exclude everybody who were poor metabolizers for CYP2C19. So for a while, the trial was off because we weren't sure we could financially and clinically get to do that. There is now a point-of-care test for this. We're not sure how great it is, but you could do this. We did this in Euclid. I will speak to some of the controversy after the trial as to whether or not that had any effect on the results, but we did exclude patients who were homozygous for this mutation that inhibits the way clopidogrel is broken down in your liver and then how it might be effective. It's important to recognize in the prior ticagrelor trials that they had done, although they hadn't excluded those patients, they'd done some genetic analysis to show that it hadn't really TIMI major bleeding was the bleeding endpoint. It's truly global. 13,885 patients from 811 sites in 28 countries. I was talking to Jerry and others. We're still trying to figure out ways to enroll patients out of Africa and other parts of the world, but in general, we were able to get around to a lot of places in the world where these patients were. Here's how we did in getting the patients in and following them. 16,237 patients were evaluated for eligibility, so they were actually enrolled. Of those, they failed for a variety of reasons. I'm just going to highlight that 600 or so patients failed for that genetic polymorphism, the homozygous status. So not a tremendously large number of patients. We had actually done an estimate when we were talking to the FDA. Back when the trial was 11,500, we had estimated that there would be 400 patients based on what we thought the population prevalence was of homozygous status that would get into the trial. So we were arguing to them, we're going to go screen 15,000 patients to find 400 patients. All this cost, will it make a difference? They said, yes, it will. Please exclude them. So I said, okay. And that's how it goes sometimes. We randomized these 14,000 patients, and in each arm, 20 patients or 23 patients never received a dose. Withdrawal of consent was present in 1.8% versus 1.6%. This discontinuation rate here you see includes the primary endpoint in death, so it's not including the primary endpoint. Unknown vital status. Unknown vital status was present only in 7 patients in each arm. Loss to follow-up was present in 1 patient in the ticagrelor arm and 4 patients in the clopidogrel arm. I'll just say this is a testament to the tremendous amount of work that our operational teams, the AZ team, I know the DCRI teams here that were beating on to say, call the site, tell them to call, figure out what's going on. This is a measure of quality in trials. We spent a lot of time trying to make sure we got all the data. Complete ascertainment of the primary endpoint as measured as a proportion of the potential patient years with follow-up. This isn't routinely described. We described it just to think about what is the total number of patient years that was possible, how much ascertainment do we have? Again, pretty high, over 98%. Here's what the patients in Euclid look like. They look like the patients that we thought we wanted to get in with PAD. Age was 66, median age was 66. We obviously are striving to always have more women in the trials. 30% current smokers, so these are PAD patients that were current smokers. 29% had coronary artery disease, defined as prior MI, prior PCI, prior CABG. Patients with PAD had multivascular bed disease. 33% had disease in two vascular beds, meaning heart, brain, or legs, at least two of them. 10% had it in all three beds. So these are patients with at least atherosclerosis known in all three vascular beds. 38% had diabetes, high proportion of patients with hypertension and hyperlipidemia. The ABI value, we wanted 0.8, so 0.63 in the patients enrolled based on the ABI value, 0.78 for the patients that came in symptomatic prior revascularization. That means they were symptomatic, they got blood flow to their limb, and it was still 0.78. So just and says, well, this is what I think it is, this is what I think it's going to be. And I can tell you almost every time you turn the page, and it's rarely what anybody thinks it's going to be. It's why you do the experiment. It's why we want to learn. There are a lot of reasons that we do these things. I told our team we learn from these things. I'm going to tell you about all the things we've learned here, but when you first see this, you're always struck by the finding. The second thing I'll say is that this finding left no doubt. As you can imagine, once I show you this finding and I show you the next set of slides, the interpretation piece we'll talk about, but it left no doubt, at least with the testing of these two therapies. So that's the hazard ratio right around 1. We were looking for a 15% relative risk reduction. Here's the composite. So if you were to ask the executive committee before we started, what was the piece of the composite we were most at risk for not hitting, they would have said ischemic stroke. And so when we unblinded, we said the cardiovascular, the composite, this is now just the raw rates and events, similar, cardiovascular death, similar, myocardial infarction, similar, ischemic stroke, statistically less with ticagrelor. The composite with acute limb ischemia is similar between the two groups. All-cause mortality, all-cause mortality, all-cause stroke. I'm sorry, cardiovascular death, MI, and all-cause stroke. Hospitalization for acute limb ischemia, lower extremity revascularization, all revascularizations. I'll just say the hazard ratios all are near 1. Not really much to say between these two, what I'll say, effective therapies. How about bleeding? It's first to recognize that these are outpatients, even though PAD patients, as I've described to you, can be sick. They have a lot of comorbidities. We enrolled them not in the hospital, in the outpatient clinic when they were seen by their physicians, a variety of physicians, cardiologists, angiologists, radiologists, vascular medicine physicians, vascular surgeons, primary care physicians. So we went and found a lot of different places these patients could be. Bleeding was infrequent and similar between the two groups. It's worth noting that adverse events leading to discontinuation were more frequent with ticagrelor, mainly due to a described dyspnea finding and some bleeding. All of the sort of forest plots were similar except for these two that I'm going to highlight. And of course, they're hypothesis generating only, not trying to make any final suggestions, but they do tell us that some of the things we understood, so patients with a history of a coronary or carotid revascularization or patients with a history of a coronary stent at least seemed to have point estimates that were lower with ticagrelor. So we concluded that in patients with symptomatic PAD, ticagrelor was not superior to clopidogrel for the reduction of cardiovascular events. Major bleeding occurred at similar rates in patients treated with ticagrelor and clopidogrel. The clinical interpretation was, and I will state again, you do 6 years of work as a team, you want to improve human health. I believe we have, and I'll tell you about how I think we have. First is, unfortunately, we still have limited options for patients with PAD. What are the antiplatelet options we have for patients with PAD? Aspirin, clopidogrel as monotherapy. Vorapaxar, a newer drug, again, approved in a different trial we could talk about, as part of an add-on therapy. So we said there's limited options. Should we have had a non-inferiority analysis? We can talk about that. The group now sits and thinks some. I'm not sure the FDA would have thought much about it because those are traditionally shared for things, but we can talk about it. We said that we thought in this active comparator, that clopidogrel in this trial was an active comparator, and it's effective for patients with PAD. May not be as effective as we wanted, but it's certainly somewhat effective, and ticagrelor at least appeared to be comparable on efficacy and safety. We also learned potentially that when we extrapolate evidence from patients with coronary artery that. Communication, how we interface with ourselves, who our customer is, how we work with each other. I put leadership on. Sometimes there were too many times where people said, we can't do this trial until we know what Manesh thinks. That's probably not the right way to have it, and have to have more delegation and more others involved. So that's why it was really great. You can see how much fun we had, because fun is good. But Schuyler and others were learning. I remember when I was on Rocket, it was, and I'm not sure we're going to go back to those days, I know, but it was Caliph and Mahaffey and me, and Piccini was the fellow. I had like 4 or 5 people all engaged. I think we missed an opportunity here, maybe some more people or a fellow assigned. We had one early. Could we have signed others? Our bench of people that learn how to do this research is something we can work on. Data integration. So you heard about all the things we were innovating in, but I still think there's an opportunity for us to say, how do we integrate stats, ops, CC, real time? So that CC is being informed by what stats think, stats is seeing that from CC, ops is seeing all those. I don't want it to be too negative. I think these are where we do it probably better than anybody else in the world, but these are places I think we can do better. So here's some examples. We always overestimate event rates, right? So because we never had a predicate trial, we thought it was 6.5%. Around November of that first year, we started saying like, boy, we think it's 5.5, but it was really like 4.5. So we had to fix our estimate, and underestimating it or getting it right was an issue. That led, though, to 14,000 patients that we quickly enrolled. You heard about the internal database start, transition to merge, getting electronic CEC was important. Some things you can't control. We have risk-based monitoring, and then we had a site issue where the site was probably doing something we couldn't understand until we actually had them self-report a quality issue. Those are things you can't find out. Database lock. I just write the database lock story. I remember we were trying to lock on the 26th. I was in the cath lab. They're like, okay, we're ready to lock. We're locking. We're ready to lock. Everybody ready? Go sign that form, Manesh. And then like Craig and people are like texting me, get back on the call, get back on the call. So I get back on the call. I'm like, what's up? They go, a site in X country just entered more data into the database. Like, wait, how did they do that? We didn't think they could do that. They're like, I'm sorry, we're going to probably have to work again and not lock today. And it was like somebody had just punched us in the chest because we were all so ready to do it. But to the credit of the DCRI and the stats team helping the data team and the data team helping the stats team and the leadership saying, okay, I'm going to need to time myself out for about a half a minute, but then I'll be able to get back on a phone call and talk to the sponsor and say, here's what we're going to do. We were able to move forward. So what worked? Well, at the end of the day, it's what works at the DCRI all the time. It's people, right? Like, it doesn't matter what the system is. I can get on a phone call and say, Craig, we got a problem. What's the story? Or Denise, what are we doing about this? Sarah, what's the story? And people would tell me what the thing is. So at every turn, I think we worked together with each other and helped answer the problems. And one of the things that helped is at every turn we were thinking about, well, what are those PAD patients going to get if we don't figure this out? I'm still got to go back to clinic and make a decision. Is this patient going to get clopidogrel? What's the story? The other thing that really worked, you don't get 14,000 patients in 14 months unless the investigators really want to answer those questions. One of the things that happens at the end is the people have to help get the trial done, which we had happen. And I think the protocol was pretty close aligned to our clinical practice. So when I was in clinic, we could say, oh yeah, so the clinicians on the team said, yeah, I can put this patient in the study. We have to work to integrate that faster, but we often overcome problems because of our people, which is fantastic. But I'd love to keep working on that structure so we can really maximize what our people do. So I end with those randomization slide. And so what about the future of PAD? There's agents like Vorapaxar. There are other drugs being evaluated, and we have to understand how to do that against what I'll call the standard clopidogrel. I think we have to have a standard data structure that integrates research and healthcare delivery at sites. Craig will come up and maybe answer some. Chris.
Thanks, guys. That was terrific, and it's great to have both the results. More importantly, I think for this group, the kind of deep dive into the operational issues, and I do think to all your point, we still don't do a great job at DCRI in institutional learning, in taking lessons from trials and projects and making sure they're applied to the next one, rather than reinventing the wheel each time. And I think this type of research conference really helps us with that. Now, two brief questions. You mentioned this 14 patients that didn't have vital status, but how about, did that include withdrawal of consent or not? So that was including all patients who withdrew consent.
That's right. So 14 patients didn't have vital status. A lot of countries, even if you withdraw consent, you can use publicly available records to do vital status. You can do several now. We also had in some of the consent forms where a lesson learned would be on withdrawal of consent to have an opt-out for public not an opt. You have to opt in to saying, I don't want anybody to use anything instead of an opt-out. So like Harvard and others say, when you withdraw consent, we're still going to search every publicly available data source. And the only reason we won't do that is if you check this box and say, I don't want that. So we try to use some of that.
Yeah, or not have that at all.
Or not have that at all, but
if you're in the trial, you agree.
We tried that, but there are a lot of local regulatory places, at least in the United States or others, that might want that box.
And then the second that you mentioned was the AEs, which and you make the great point. Why collect? So what was the strategy for? Presumably, if it was SAEs that were outcomes, those were not collected as SAEs. Yeah, so, and were any AEs collected or not?
Yeah. So we had a conversation. This is often two-part. As you know, Chris, it's not just It's not just DCRI always, right? I mean, it's sponsor, it's regulatory groups. So first is anything that was a clinical event shouldn't have been an AE, right? So that being said, mortality around the world still gets sometimes reported, even though we were trying to get it not reported as an SAE. And then we had adverse events of interest. Not all SAEs, but adverse events of interest, which included dyspnea, because we knew that could be an issue, gout, actually potentially for ticagrelor, and then unstable angina, atherosclerosis in the carotid, atherosclerosis in the lower extremity. Those were going to be called disease progression by the CEC, and hopefully not have to be reported as SAEs. We tried to build that loop. That loop probably has more work to be done as a group. Even then, at the end of the trial, there was such fear that if this agent was going to go for review, that if we missed an SAE at a site, we were going to be just held accountable. So we were, scouring these things to get SAEs at hospitalization, and I was asking these questions again, saying, I recognize we have to do it, but if we have the right patients in and we get the endpoints accounted for, what do we do? This is a cultural thing, as you know. So again, yeah.
Question.
My staff colleagues, some good questions. I'm going to defer all of these to
well executed trial. One thing I need to add is, for full disclosure, I was
The DSMB member.
DSMB. So there was one interim analysis, and during the interim analysis, we really struggled why the leadership and the sponsor didn't want futility stopping for the study.
So, so glad it didn't happen, but I can talk about that some. So, Hussein's asking a really great question. He was a statistician on the DSMB saying, they really struggled saying, why was futility not allowed in this study? And we had actually a unique thing happen. When we did the study, we thought it would be that the interim analysis, when the interim analysis would happen at halfway point, that it really may be farther than halfway just because of the way the data cycle is in getting patients in. So we were as an EC pushing to say, we just don't want a futility analysis because we don't think we'll be that far from the end. That being said, every month is financial, and I understand there's reasons, but we didn't know, because of other histories, how far along would the futility analysis be and what would the boundaries be. Right? But there are some defined boundaries, and you could have applied them to Euclid and potentially said, we've reached those boundaries. We just did push not to with the EC because we thought, with the sponsor, because we thought we were going to be 6 to 8 months from trial end. And we said, if we're 6 to 8 months from trial end, even though we recognize there's an opportunity to make a very sound futility analysis, there's a lot that can be learned. And thankfully, they agreed to that. We can all agree whether or not. The other thing I'll just say about this is a futility analysis, but it's in an active comparator trial. And so patients in the trial are getting therapeutic in both arms. You can decide if that's important or not.
So Manesh, great presentation. Thank you. What are your thoughts now about why there was not a difference between these compounds in PAD, but prior experience in the setting of acute coronary syndromes showed a different result? What thoughts do you have on the explanation for that?
I sort of have two. One is the available data is that, I'll just go through the existing data for ticagrelor. It's better with aspirin compared to clopidogrel with aspirin when somebody's having a heart attack. That's an activated plaque, an acute event. It's better with aspirin compared to aspirin alone in a stable patient with coronary disease. There, I think aspirin's the comparator and it's a more potent drug. What we've shown now in a PAD population outpatient with atherosclerosis, but maybe more stable than the first two populations I've described, or a different population, it's very similar to clopidogrel. So my view of that is, it could be that in patients that are acutely ill, or patients with recent coronary events, there's a benefit where the higher potency helps. Or it's obvious, and in the stable population, there's what we'll call a threshold effect. Once you have enough inhibition, these patients do okay. And this trial tested them in the best scenario, where all these things are set up to where it's a real clean test. The second is, of course, that the PAD population is different, and their cardiovascular event rates and drivers of cardiovascular events may not be direct plaque rupture, but maybe a set of other things. Those two biologic questions we don't know the answer to. What we obviously know is that the findings from the trial are very similar. The CYP2C19 homozygous question, because of the way the findings are, for this trial to be positive for ticagrelor, all six, we would have had to have a 70 to 75% relative risk reduction in those patients that came into the trial to have found a difference. So I don't think that accounts for it, but I don't think it, obviously, I think it's just the nature of what we studied. And we didn't know that, right? We thought more potency is better. What you probably need is another pathway in these patients. This pathway is covered enough if you do this. There is. So the ischemic stroke data is being pooled. The drug is being studied in an acute stroke trial, and I can tell you in other ways there's a huge need for acute stroke, and ticagrelor may actually have a role there. They're actually going to do, I think, another type of study in acute stroke. They've done one with this data and the others. I think there's an opportunity for patients with ischemic stroke. There, the biology is probably different again, but there is an activated thrombotic action. All right. Yeah, one last one, sorry, or we'll let people go.
Related question to Dr. Lee's question regarding ischemic stroke. So we know for the Socrates trial that ticagrelor is not as good as aspirin, but for Lucid, we see that's a better outcome among patients taking ticagrelor than Peripheral. So although they are not the same comparison, but what will be your thoughts on this?
So in acute ischemic stroke in Socrates, what we know is ticagrelor compared to aspirin gets really, really close, and in some groups would do it. And in this group, monotherapy in PAD patients, ischemic stroke is reduced. So I think ischemic stroke is an opportunity, both a population and an outcome for ticagrelor that might be beneficial. Okay, thank you all. I'm going to be representing time.